I'd like to welcome our
Perfect for the
second
time.
speaker for this session, Romain Boideau. Romain is a molecular biologist at Centre Georges François Leclerc in France, and he's been there for 12 years. He's in charge of the molecular biology unit dedicated to routine molecular diagnosis related to solid cancer. He's also responsible for a next-generation sequencing service platform. Thank you.
Thank you. So I want to thank the organizer to give me the short opportunity to present the potential use of nanopore sequencing for routine molecular diagnosis of germline cancer predisposition. The current analysis uses only coding regions, single nucleotide variation on large-scale rearrangement on 2 to 3 dozens of genes. And by analyzing these genes, we are able to detect alteration only in 20% of samples. So more than 80% of samples do not have any alteration detected. So we have two different solutions to give an answer to more than 80% of patients. The first one is we can analyze other genes, either by exome sequencing or whole genome sequencing, but both technologies are not yet applicable in clinics. We have also the opportunity to detect non-coding region of already known genes, and for that we can use Oxford Nanopore technology to detect DNA methylation, DNA modification, and the complete gene sequence from promoter to 3' UTR. And this technology is quickly applicable in clinics. So the objective of our project is to demonstrate the ability to detect clinically actionable variants from adaptive sampling for inherited cancer. And for that, we set up different steps, from DNA extraction to DNA input to engage for library preparation, the sequencing protocol to improve sequencing yield, and especially by adding a wash and reload step. We developed the bait file for adaptive sampling and fired bioinformatics pipeline. And as you can observe on this quite complicated graph, we started our experiments with a large bait file representing more than 4% of the genome, and we obtained a quite low coverage of targeted genes. So we decreased the bait file, and by decreasing the bait file, we did not impact the sequencing yield, but we increased the coverage of target genes. We were also able to improve the amount of libraries that we were able to put on the flow cell, and we also increased the sequencing yield thanks to this increasing quantity of library preparation loading. And finally, we systematically perform a wash and a nuclease wash and reload library preparation for each run. So as you can observe, we can easily visualize single nucleotide variation thanks to the Nanopore sequencing. It's quite clean. And regarding structural variation, on the left side, you can observe a patient with exon 13 duplication detected by MLP and BRCA1 gene. And by using Nanopore sequencing technology, we're able to detect and confirm the duplication of exon 13, but we're also able to detect a duplication of exon 12. And in fact, the duplication was a duplication of exon 12 and exon 13. And on the right, you can observe that an heterologous deletion of BRCA1, and this complete heterologous deletion of BRCA1 will also bring 5 other genes. So just to finish, I wanted to thank Sandy Chevalier for the wet lab experiments, Corentin Richard for bioinformatics help, and the sequence genomics team for bioinformatics analysis. Thank you for your attention.
Thank you, Romain.